Enormously excited to welcome back Avery August from Cornell University, formerly of Penn State. But once you're part of the family, you're always part of the family. So the floor is yours.
Thank you, Matthew. Can you all hear me in the back? Great. Well, thanks for the opportunity to talk a little bit about some of the stuff that we've been doing, and it's going to be a little bit different, although you've heard a bit about sort of being inside the host earlier. I also want to pick up on a thread that Samuel talked about with regards to tolerance and resistance. It'll become clear as I go through. But thanks to the organizers for really inviting me back. I always say yes whenever anyone says, come on down. So thanks again. So wherever I am, I always try to acknowledge where I am. So I'm in the traditional homelands of the Munsee, who were an indigenous Delaware and Indian nation here in this region. Also disclose that I do have some funding from 3M company. I'm not talking about that work today. So the work that I'm going to talk about is sort of 2 or 3 vignettes, sort of try to illustrate a point I want to make that picks up on that thread of tolerance and resistance. And the work is done largely by a number of graduate students in the lab, starting with Brian Ibiaka, who's defending in a couple of weeks, with Julius Taylor, who's a senior research associate, Arun Kannan, a former PhD student, and then Wei-Shen Wang, who actually started her graduate work here and moved with me to Penn State, to Cornell, and now is at LSU, and with help from Jessica and Amy. So I want to pick up on this idea of the importance of the immune response. And we heard Yasmin talk about the adaptive immune response in terms of interacting with the microbiota. And I want to step back a little bit and simplify this a little bit and just sort of refer to the adaptive immune response and the innate immune response. Of course, the innate immune response is sort of more primitive, evolved earlier. And in its absence, if you look at microorganism count here, because they're not able to control microorganisms through simple mechanisms like phagocytosis and other things, you end up with increasing numbers of microorganisms and eventually potentially death. If you lose the adaptive immune response, the innate immune response is able to sort of control microorganisms better in its presence, but at some point, you're not able to reduce the microbial burden, and in some cases, in many cases, you can lead to death. The normal, of course, response is you have an infection, the number of microbes increase, both adaptive and innate immune response kicks in, and you clear the microorganism, and so you survive. So I wanted to start with some work that we've been doing using SARS-CoV-2 that challenges that a little bit. And that's, sorry, I just wanted to just quickly remind everyone how the adaptive immunity gets activated. Microorganisms are recognized by patterns, by innate immune cells. These then traffic to the lymph nodes and hand off those signals to adaptive immune cells, such as B and T cells. You get T cell responses, you get antibody responses. So the first story I want to tell you, a short story about some work that we've been doing in collaboration with Hector Aguilar-Carreño at Cornell on trying to understand the interaction between SARS-CoV-2 and the immune response. So we've been using a number of different SARS strains, using mostly mouse models and other models, both the original strain as well as the evolved strain as they've evolved over time. Most recently, we've been using a strain that was developed most recently by List et al., which is a mouse-adapted form of SARS-CoV-2 that allows it to infect wild-type mice. So the mouse models that many of us have been using to study SARS-CoV-2 is a model that overexpresses the human receptor for SARS-CoV-2, allowing it to be infected. And so that has some challenges. And so more recent models have evolved that use either a knock-in of the human ACE2 receptor that allows these mice to be infected with the human virus, or evolving the mouse strain to adapt, the SARS strain to adapt mice. And so that's these experiments I'm going to tell you about. So we use this mouse adapted strain called M810. And what we did is we infected mice in BSL-3 facilities. And what we found, to our surprise, is that while wild-type mice are susceptible to infection, that is, they lose weight, Some of them die, but we try to tune the infection so that most will die because we want to study their immune response and then recover. What we found was that mice without an adaptive immune system actually don't lose any weight at all. And so that was really surprising because I just told you that the adaptive immune response is really important for eventually clearing infection and restoring host health. And so that was surprising to us. And so it sort of suggests that this model of the adaptive immune response being important for eventually clearing this particular virus, may be a little bit more nuanced than this model suggests. So we actually looked to see, are these mice actually able to clear the virus? And it turns out they're not able to clear the virus. So you see here, these are infections that have gone out to 30 days after infection. The mice are perfectly fine, no weight loss, they look fine. Remember, they have no B or T cells. The wild type animals that recover, they clear the virus, whereas the animals that lack B and T cells are not able to clear the virus. And that's either looking at viral copy or actually plaque forming units of the virus. If you look at immunopathology, you take sections of the lungs from these animals, you see here that the wild type animals have lots of immunopathology in the lung. So the immune response is happening. The adaptive immune response is actually generating a lot of activity against the virus, it's clearing the virus, but it's also causing a lot of immunopathology. Whereas if you look at the lungs of the animals without B or T cells, they actually have very little immunopathology, again, indicating that the virus is able to replicate, but because of the absence of B and T cells, they're not causing any disease. So this model of the function of the adaptive and the innate immune response, while true, perhaps is also nuanced with regards to what microorganism we're looking at. So in the case of SARS-CoV-2, and maybe this particular strain, that the adaptive immune response is actually important, and in experiments I don't have enough time to tell you, it's important for enhancing viral replication in the lung, because early in infection, the mice without B or T cells have lower virus, but later on, the wild type strains clear the virus, and then the animals without an adaptive immune response sort of maintain that level of virus throughout the time that we've looked at them. So in other words, the adaptive immune response in this case is both important for clearing the virus, but also contributes to immunopathology that can lead to death. So the adaptive immune response then is critical for the protection against the viral infection, but also this sort of idea of being able to contribute to immunopathology. So we wanted to look at another virus to see whether that's the case. And some old work that was done by Cassiotis and his colleagues looked at flu infection with the same B and T cell deficient mice that we've used. And what they found was that if you infect these mice with flu, they die. So it's not because it's a viral infection. It's a particular type of interaction between SARS-CoV-2 strain that we've been studying and the immune response, such that the adaptive immune response actually plays an important role, both in enhancing viral replication, but also clearing the virus, but in causing pathology. So that's one story I wanted to tell you, and I'm happy to answer questions later on that. The other story I wanted to tell you is trying to understand now the host response, particularly adaptive host response to pathogen infection. And what we've done is we've taken a mouse model where we've weakened the T cell response by mutating a protein that's important for activating T cells. So you still get an immune response, but the T cell response is weaker because the T cells get a weaker T cell activation signal. And so we've been studying, in this case, flu infections using this model. And here's the sort of simple experiment where we infect with flu, and then we study over time the behavior of the animals. And what we found was that when we reduce the ability of T cells to be activated, we actually get these animals don't survive very well, whereas the wild type animals are able to survive. Now you might say, well, of course, you've weakened the T cell response, so therefore these animals don't have a strong response against the virus, and so they're dying. But what was interesting is when we looked at the amount of virus in the lungs of these animals, there was no difference in viral clearance, and eventually both strains will clear the virus. So this response that we're seeing when the T cell response is weakened and the fact that these animals are dying is not because they're not able to generate an immune response and clear the virus. It turns out when you look closer in the lung, again, if you look at over time, 7 days up to 11 days, the wild type animals that are infected generate a certain amount of immunopathology. But if you compare here, for example, 11 days after infection, these animals have much higher levels of immunopathology in the lung. So in weakening the T cell response, we actually have generated a case where we've increased the immunopathology, allowing clearance of the virus, but then the animals die. So tuning the adaptive immune response seems to be important. And actually, if we look a little bit closer at cytokines that are reflective of an adaptive immune response, what we see, these are neutrophils, so inflammatory cells. We see a lot more inflammatory cells in the animals that have a weaker immune response, a weaker T cell immune response, and are dying and clearing the virus, indicating that they're generating very strong immune responses against this particular infection. And if you look at interferon gamma, a very classical a cytokine, Th1 cytokine that's important for clearing infection, you see that there's no difference in the abilities of these animals to generate interferon gamma. But what was really striking to us is when you look at interleukin 10, which is a cytokine that's actually an immunosuppressive cytokine, they actually generate a lot less of interleukin 10. So what we're doing here in weakening the T cell response is we're starting to tune the type of immune response that's generated to this virus. And so these animals are able to make interferon gamma and therefore clear the infection, but they also make less interleukin 10, which is supposed to dampen the immunopathology. And so while they clear the infection, they die. So looking back now at this simple model, thinking about the role of the adaptive immune response, if we're weakening this adaptive immune response here, we're now start to see tuning within the context of the antiviral response such that you can still get clearance of microorganisms, but you also get death because of imbalance of immunopathology. So the other example I want to give you is another model that we've been looking at in working with Eric Denkers, who studies Toxoplasma gondii. And toxo, again, requires a very strong interferon gamma response for clearing. And again, similarly, we wanted to examine what would happen if we weaken the T cell response and infect these animals with toxo, would we get a similar response? And what we found is that similarly with the flu, when we infect these animals that have a weaker T cell response with cysts that generate an infection, we actually see that the animals that have a weaker T cell response lose less weight over time, but once they start losing weight, actually they die, and they start dying by 30 days. So again, they're able to survive a little bit better than the wild type animals, but eventually these animals die during the immune response to infection. And so coming back to this idea of how the adaptive immune response is interacting with these with microorganisms, the model here is that there's a balance that the T cells have to maintain, and that if that balance is weakened, we disrupt the homeostasis of that immune response. So we're able to get interferon gamma being made, but not able to control immunopathology. Indeed, if you look at the clearance of toxo infection in these animals, they're actually better at clearing the toxo infection. This is now looking at genome copies by PCR in a variety of tissues of these animals. You see that the wild type animals is still some residual microorganism, particularly in the brain. But if you look in the animals that have a weaker T cell response, they actually have cleared or largely cleared the infection. So they're able to generate this immune response through this process of pattern recognition, generating T cell responses that is able to make interferon gamma, but that cytokine production, while sufficient and able to combat the microorganism, needs to be balanced. So what is that balance that's required? So I've been telling you about the sort of generating of the immune response through these two infections, and that immune response is largely activating of naive T cells that generates Th1 responses, and those Th1 responses make interferon gamma, and that interferon gamma then is really critical in helping to clear the infection. And what we've done is we've weakened the activation of these T cells, which still allows the cells to make interferon gamma, but affects other parts of this response. And the other part of the response, this is just describing that both flu and toxo require interferon gamma for their clearance. So if we look at the interferon gamma production in the toxo-infected animals, what we see over time, this is now looking at serum levels of interferon gamma. What you see is that, again, the animals that have weaker T cell responses actually have higher interferon gamma levels than their wild-type counterparts, and they start dying around here, around 30 days. So again, they're generating an immune response, they're controlling microorganism, but they're not able to survive much longer. So this idea of infection leading to an immune response is an important one to think about, but also thinking about the impact of the immune response that's generated by that infection on pathology, and particularly immunopathology. And in this case, interferon gamma in both toxo and flu infections is critical in doing that. And so there has to be a mechanism to, once the immune response is turned on, to turn it off. And one of those mechanisms is the generation of what are called T regulatory cells. So these are these other effector cells that can also be generated during an immune response from the same precursors. And in this case, there are two types of T regulatory cells that have been described. These are FOXP3 positive T regulatory cells. They express a transcription factor called FOXP3, and another type of T regulatory cell called a type 1 regulatory cell. In both cases, while they're generated through different mechanisms, in both cases, they make interleukin 10. And that interleukin 10 is really critical in balancing the immune response that's being generated. So these T regulatory cells then are also generated during infection. And in addition to generating the sort of effector cells that are going to be clearing the infection, the immune response also generates these T regulatory cells that can make interleukin 10, that can modulate the pathology such that you can control the infection while also controlling immunopathology. And so if you don't have cells that are making interleukin 10, when we weaken the T cell immune response, we generate a scenario where the cells are still able to make interferon gamma, but they're not able to make interleukin 10, even though they still have these regulatory cells. Now you have immunopathology, and while you clear the infection, you also end up suffering from that. So you then get runaway interferon gamma. So we wanted to study this a little bit more at the cellular and molecular level. And so we generated mice that allows us to follow the generation of these cells over time and space. So what we did is we crossed mice that express a GFP, green fluorescence protein, whenever they're making IL-10. So we can follow them over time. And also mice that express red fluorescence protein whenever they're generating these FOXP3 Tregs. So we can follow both the generation of the FOXP3 positive T regulatory cells, as well as other T cells that are also making interleukin 10. And we can follow this by using flow cytometry and microscopy methods to be able to follow these cells. And so when we looked in flu infection to see which cells are making IL-10, what we found, and I'll just go through this really quickly. So these are flow cytometric plots where we're gating on different populations of cells from the lung after flu infection. And the relevant places is to look here, where when we gate on cells that are expressing IL-10 because they're expressing green fluorescence protein on the y-axis, and these are T cells, what we find is both CD4 positive T cells, but also CD8 positive T cells. These are the cells that are actually supposed to be clearing virus, are also making IL-10. So the same cells that are making interferon gamma and also killing virally infected cells can also make interleukin 10 and suppress that immune response. As a matter of fact, if we do this the same experiment and sort of combine a number of different animals, what you see in just focusing on the flu infection here is that the cell types that are making IL-10, a large proportion of the cell types that are making interleukin 10, this suppressive cytokine during flu infection, are actually CD8 positive T cells. And then, of course, the other remaining cells are CD4 positive T cells, either FOXP3 cells, actually a very small proportion of the FOXP3 positive T cells are actually making IL-10. The bulk of the other CD4 cells that are making IL-10 are these type 1 regulatory cells. So the cells that are involved in generating the immune response against the virus that's generating sort of the inflammatory environment are also the cells that can also generate the environment to sort of suppress that. And when we looked at CD8 positive T cells more carefully, again, during flu virus infection using flow cytometry, what we see, again, looking at cells that are making IL-10 over here, and these are now looking at CD8 T cells. What you see is that 7 days after infection in the wild type animals, about 60% of the CD8 positive T cells are making IL-10. And this is sort of at the peak of the antiviral response, at the peak at when the cells are actually starting to generate immunopathology and killing virus. And if we weaken the T cell response, we actually get a lower proportion of those cells making IL-10. Similarly, if we look at CD4 cells, we see a similar scenario. Again, focusing on day 7, you can see that wild type cells, CD4 cells are making IL-10 here. And if we weaken the T cell response, we actually get less IL-10, and that's quantified down here. So we wanted to study this a little bit more. And so we generated some in vitro systems to be able to differentiate these cells. And we could show that this was a cell intrinsic effect, because if you just isolate those CD4 cells and differentiate them in vitro, you can get cells that can make IL-10 over here. And if we weaken the immune response over here, we get a lot fewer of those cells. We also asked what would happen if you actually inhibit this pathway and actually reduce T cell activation using a small molecule. And you see a similar situation here where we use a small molecule compound and we can reduce the ability of these cells to make IL-10 over here. So, so I told you that during flu infection, these animals are able to make interferon gamma, regardless of whether they have a fully functional T cell response or a weakened T cell response. But when they have a weakened T cell response, they don't make as much IL-10. And that balance is really important for controlling immunopathology, because without controlling immunopathology, they're able to clear the infection, but then the animals die. And of course, this CD4 positive T cell response is really key to doing that. So in this case, we're generating cells that can make interferon gamma that's sort of contributing to clearing the virus, but at the same time also making these immunosuppressive cells. By weakening the T cell response, we're able to generate these cells, but not these cells in the same way. And so we wanted to determine whether we can manipulate these early signals and manipulate the type of immune response we're getting. In other words, can we change the nature of the T cell response by weakening the T cell activation and maintain and change what these cells become? So instead of becoming TLP1 cells, are these cells able to change what they become? And so we did a simple experiment where we took naive T cells and cultured them under conditions where they would normally become T helper 1 cells. So these are the cells that are making interferon gamma, both in the toxo infection and in the flu infection. And then we reduced the activation of these cells by inhibiting the pathway, and we asked what happened. So I'm going to take you through that experiment shown over here. So again, we can take wild type cells and we can generate cells that can make IL-10. This is what they would normally do. We also measured interferon gamma, which is the sort of inflammatory cytokine. What we see here is when we genetically reduce the activation of these cells, the cells no longer make IL-10, but quite surprisingly, what we find is that they're actually making more interferon gamma. So by reducing the ability of these cells to be activated when they see antigen, we actually skew their response to a pro-inflammatory response as compared to an anti-inflammatory response. So in other words, normally they would make both IL-10 and a proportion of them would make interferon gamma, and that will be the balance that you would see in vivo during the immune response. But when we reduce the activation of these cells, we actually don't get the IL-10, we actually get more interferon gamma, and so therefore we can see the effects of that immunopathology. We can actually mimic that also with a small molecule that also inhibits this pathway. Again, we're reducing the ability of these cells to make IL-10 and increasing their ability to make interferon gamma. So what these experiments suggest is that within the T cell response, one can tune their responses by manipulating the ability of these cells to be activated. In this case, we use either genetic means or an inhibitor. And normally these cells will become TR1 cells and are making this IL-10. But when we inhibit the activity, we switch these cells and now they make more Th1 cells, they make more interferon gamma. And so now they're able to clear infection, but then because they have so much interferon gamma, which is not balanced by IL-10, then these cells, these mice don't survive very well. So, so I told you then that when we think about the interplay between the adaptive immune response and clearing infection over time. What I showed you was an example of a SARS-CoV-2 strain that is able to infect these animals and doesn't kill the animals. The virus is able to replicate and remain as healthy as we can tell. Whereas when we use flu infection, you actually need the adaptive immune response to both clear the virus and control the health of the animal. And then finally, toxo behaves just like flu in that you require the adaptive immune response in order to both clear the infection, but also control immunopathology. And the reason that you need the adaptive immune response to do both is because we have cell types that are differentiating during an immune response, these T cell cell types that both generate the interferon gamma that's required for clearing infection, but also cells can be generated that are regulatory cells that will suppress pathology, allowing this infection to be a healthy infection that is generate enough tissue damage to clear infections, but not enough that these animals are able to die. And so when you disrupt the activation of these cells, such you only allow the inflammatory compartment to be activated, but not the anti-inflammatory compartment to be activated, then you get immunopathology and death. I also told you that we can actually manipulate these cells in vitro using small molecules so that we can manipulate these cells from becoming TR1 cells and enhancing T helper 1 cells and interferon gamma responses. And so with that, I'm going to stop and hopefully there's time for a few questions. I want to thank my colleagues, Eric Dankers, Steve Dewhurst for the MA10 strain, and Hector Aguilar Carreño for collaboration with the SARS-CoV-2. Thank you.
Excellent. We have some time for questions. Right. Oh, I was going to use the moderator's prerogative, but
thank you. This was really, really interesting. So I was wondering, are there any pathogens that would target specifically this regulatory versus effector T cell response that uses it as a mechanism to basically invade the host?
Yeah, so it's a great question. It turns out that there are a number of parasites, parasitic infections that actually enhance these cells that make interleukin-10. And by generating interleukin-10, they sort of suppress the immune response so that the parasites can survive better. There are also viruses, like some of the DNA viruses actually encode viral homologs of IL-10, and that allows these viruses to then maintain their presence in the host.
Girish?
Yeah, great talk, Avery. My question is that in the absence of ITK...
Is your mic on?
Closer.
Can you hear me now?
Yeah.
Yes. In the absence of ITK, downstream of TCR signaling, right? What controls the IFN interferon gamma versus IL-10 shift? Is that down-regulating TBET or increasing CMAP, one of those transcription factors there?
Yeah, I sort of glossed over that because this gets into the cell and sort of the cell signaling. But what we found is that the same T cell receptor pathway is used in both of these cell types. But if the cells use a different set of transcription factors to turn into those IL-10 producing cells as compared to the Th1 producing cells. And we can actually show that by disrupting the pathway that leads to the transcription factor, particular IRF4, you start getting the Th1 cells and not the TR1 cells. And you can reverse the system by putting back IRF4.
Now I'm going to use the moderator's prerogative now. We have on here? I'll let you do it. I'll ask in the discussion.
I was just wondering for toxo, I was thinking about the reactivation of the cysts in the brain. I know you showed that there's a reduction of the cysts in the ones that lacked some of the T cell. I was curious if long-term, if you were to look at the reactivation of some of those cysts in the brain, it maybe would be the opposite of what you're saying, and the cysts would be more likely to turn into like tachyzoids with those deficient mice.
let me see if I understand your question. So you're asking if those cysts can be reactivated later on, long term?
Yeah.
Because yeah, that's a good question. I don't think we've probed the system to ask that particular question. So we've gone out. I want to say about 45 days when we do those when we look for cysts in the brain, but we haven't perturbed the system to ask what happens if that's reactivated.